Okay, we're going to invite our next panelist speakers up. Dr. Zhou, Dr. Yong-Hui, and Allyson Berent, and Dr. Liz Berry-Kravis. They will be talking about the recent exciting news that we announced about a large NIH grant to advance a novel CRISPR-based gene editing delivery platform at Yale University. They are part of the project team named under the grant, along with Jennifer Panagoulias. This is really a remarkable example of cross-collaboration, and in my years of working in patient advocacy and NIH, I've never seen a model like this. So we should be really proud, and I'm really excited to hear more about it in this presentation.
So I think I'm going to start right. I will start and then pass it off to you guys. Okay. Can we have the slides? Great. So what an incredible honor to stand on stage, not just representing our community, but actually representing a drug development program. So this is really exactly what FAST has been trying to do since its inception, and this is really a milestone that's quite unbelievable for our community. So I think we should all be very proud of the fact that patient advocacy is actually taking things from benchside to bedside, and this is just a true example of that. So we are a team working together, and we're going to talk a lot. Yesterday and the day before, we did talk a lot about CRISPR technology, and so I'm not here to explain that technology to you. Yong-Hui and Jiangbing will do that, but we just want to show all of the steps of drug development for such a technology. We have lots of disclosures, here they are, that you can read. And so ultimately, this is a graphic you've seen throughout the entire weekend. And it's an important graphic to understand how we can take something from early discovery through foundational NIH and other nonprofit research funding efforts, and even some for-profit funding efforts, and go from discovery to preclinical, which is proof of concept, which we talked about yesterday, and take this idea, put it in an animal model to show actually this can work, and is there early signs that it's actually safe? And then the next step is to say, is this possible for humans, and can we develop this drug for humans? And that's where we say we have to get to a human candidate. And once we get to that human candidate, then we need to understand the safety profile of that, which I'm going to talk about. We have to develop endpoints and biomarkers in order to run these clinical trials and design these clinical trials properly. And that's through the efforts of the ABOM, which is that pre-competitive effort that we talked about yesterday. And then, of course, the community effort. We need all of you. We need you identified. We need to know where you are. We need your enrollment in the global registry to really have a good sense of where these trials can run. And then finally, we need to run the trial, which we are actually funding the infrastructure to create places like Rush with Dr. Berry-Kravis, who can run these trials that are very innovative, very complicated, and we have the confidence in the team to be able to do that with something novel like this. And so the team at Yale is going to come up and talk to you about the first part of this ecosystem, which is preclinical and drug discovery.
Great. Thanks, Allyson, for the great introduction. My name is Jiangbing Zhou. I'm a professor at the Yale Medical School. Really great pleasure to be here. On the team, my role is an engineer. So another way to say my role is that I'm an Uber driver. So my role is that to deliver the payload or therapeutic modality, this case is a CRISPR, to the right cells with neuronal cells, and to the right place, which is the brain. So that's what I'm doing. So I'm very happy to be here to share the journey, how a collaboration between an engineer with biologist, with Professor Yong-Hui Jiang, who is a lead expert in the field, to lead to this potential clinical translation. So as I mentioned, the therapeutic modality we need to deliver is called CRISPR. So CRISPR actually is a defense system developed by bacteria to prevent viral infection. So in this case, we want to borrow the system from bacteria for human use. So in this case, we want to use a CRISPR to relieve the brake on paternal chromosome to activate the UB3A gene expression. So for this specific application, we want to deliver a system with two components, which including a Cas9 protein, as shown in the cartoon gray, and also a guard RNA, as shown with orange and red. So basically, the guard RNA can guide the Cas9 protein to the right location, and the Cas9 protein can cut the antisense sequence. So this is a two-component system, right? So as I mentioned, my job is to deliver the system to neuronal cells in the brain. So there's two major challenges. First, how to deliver the system into neuronal cells. We have talked about delivery in the morning, right? Naturally, you may think, why not use virus? Of course, AAV virus is very powerful. It has been probably the most popular used system in current clinic. So however, unfortunately, virus may not be the best approach for deliver our CRISPR system for two reasons. Firstly, this CRISPR system actually very, very large. In terms of protein, it's probably over 160 kilodalton. You cannot fit one machinery into one single virus. Of course, you can potentially use two viruses, but the more virus you use, the more potential side effect you will have. So another potential limitation is that in this case, we deliver a bacterial machinery, right, as I mentioned a moment ago. So virus delivery has an advantage in terms of persistent expression, right? So that's good for gene replacement therapy. But this could be a problem in this case because we want to express a bacterial protein in cells for many years. That could be dangerous, right? This protein belongs to bacteria. And also the Cas protein can cut your genomic DNA, probably in certain cases even without a guide. So you may worry that if long-term persistent RMP or Cas, right, can cut this gene which is important to you. So as an alternate approach, you can potentially use nanoparticles. So in this case, nanoparticles could be synthesized using synthetic materials which can mimic a virus. So probably nanoparticles has a terminology new to some of you, but actually you may use particles because for COVID-19 vaccine, both the Pfizer and Moderna vaccine actually use nanoparticles. So this comes to the second challenge, because we want to deliver the machinery into the brain. So as Dr. James Wilson mentioned this morning, the brain possesses a unique organ called blood-brain barrier. Or another way says that the vessel in the brain is very unique. Biologically or physically, it's very, very unique, very different from vessels in other organs. So as a result, it's very difficult to get any therapeutic molecules to penetrate blood vessels and get into brain tissue. So one way to bypass the blood-brain barrier is to inject a therapeutic molecule into CSF. That's actually what we are doing for the ASO trial, right? We inject an ASOs into cerebrospinal fluid called CSF. So the hope is that these therapeutic molecules can diffuse into deeper brain through private cellular space, which means that between cells in the brain, there's certain space. You can, in some cases, the therapeutic molecules can diffuse like an ASO. So one of the reasons is that ASO is very small. They can diffuse between cells and get into deeper brain. But the problem is that for nanoparticles, and actually also true for most viruses, they're probably too large for penetrating or diffusion through the paracellular space. Because space, as shown here, typically about 10 to 30 nanometer. AAV vectors actually about 25. Probably that's one of the reasons we can see AAV get into certain space, but not the entire brain. How to overcome this problem? We come to an engineering approach we call STEP. So in this case, for whatever payload to be delivered, of course, CRISPR, and a specific machinery we call RMP. So we can do chemical modification. So what means is that we can put a molecule we call STEP on the surface of RMP. So this STEP molecule is very powerful. They can bring this RMP into cells without being trapped in other unwanted organelles in the cells. And once get into cells, the certain molecules within cells can cleave STEP molecule from RMP, and then turn RMP into their native form, so it can start to work. The beauty of this engineering approach is that after engineering, the resulting product is very small. is about 10 nanometer. As I mentioned, right, extracellular space is about 10 to 30 nanometer. So in terms of size, they're perfect for diffusion in the brain. And of course, this step enable give some other properties, making this RMP particularly suitable for brain penetration. Of course, we need, before we get into CSF-based diffusion, we need to test these particles in cell culture, in vitro, and also in vivo. So in this slide, we just left, of course, we're testing a system called AR9 report mice. This mice, in typical sedation, don't express any fluorescent protein. What it means is that if you get cells out, if you check the cells under a fluorescent microscope, you essentially cannot detect cells because these cells don't express fluorescent protein. But if editing happened, you will see red fluorescent protein. So as shown in the left panel, we found that one single treatment can essentially turn on RP or red fluorescent protein expression in almost all cells. We also test this step RMP in mouse brain. In this case, we inject a step RMP into the left side, left hemisphere. You can see that control mice, we didn't see any red fluorescence. But with the step engineering, we can see a widespread red fluorescence. And also, unique thing, we choose a unique step molecule here. This editing preferentially happening in neuronal cells. So the right panel just shows this system is not only just efficient, but also very fast. So by 2 hours, this machinery gets into the right place, nuclei, so it starts to work. Of course, we need to demonstrate you really can deliver this machinery into the brain, right? As I mentioned, we want to do CSF, cerebrospinal fluid delivery. So this was this experiment done in mice. So step RMP was injected into CSF through lumbar puncture. So you can see that by hour 1, we can see step RMP dispersed in several different organs, spine and the brain. And by hour 6, we can see the step RMP penetrate brain tissue. And then by hour 12 or 24, we cannot detect this Cas9 or the RMP-based CRISPR machinery in the body anymore. So this could be good, right? As I mentioned, this is bacterial machinery. We really don't want them to stay in body for a long time. So this could be ideal. But of course, the question is that whether this short-term exposure is good enough, really good enough for treatment of Angelman syndrome. So over the past 2 years, my lab and also together a team from Courage, a Yale startup, we synthesized a variety of step RMP and sent to Yong-Hui's lab, and then Yong-Hui's lab tested this step RMP in mice, which mimic human AS patients. So Professor Jiang is going to tell you the findings in animal models.
Thanks, Jiangbing. While I was sitting there, I just realized Jiangbing is probably only biomedical engineer among this community. If any other biomedical engineer here, you can raise your hand. So therefore, I've got welcome the biomedical engineer to this amazing community. He is the inventor. I'm the user. That's how the relationship goes, because without inventor, we will not be able to stand in here, do this amazing work. So he has many patents for sure. I have 0 patent, so that's. I used to take a long time, like 5, even 10 minutes to describe this slide many years ago, but now with the ASO, many of you, almost every one of you are very familiar with this concept. So the idea is you can use ASO or drug to reactivate the gene from paternal chromosome. You can do the same thing too by CRISPR new technology. And the beauty for the CRISPR is you may only need 1 dose, 1 intrathecal or spinal tap, then fixed and permanent. So that's our hope is. So the idea is use a CRISPR knockout or inactivate the antisense from paternal chromosome and reactivate the gene from paternal chromosome, so contribute to the therapeutic benefit. So therefore, that's the kind of shortening for that one. So we indeed are using human neurons, which is many of you contribute the iPSC cell we generate at Yale. Then we differentiate to the neuron. We put a step, this new technology, and Jiangbing invented. Oftentimes, just to make sure, again, after we did a talk on the Thursday, after talk, people came to me and say, okay, this is a nanoparticle. So to make it clear, it's not viral, not nanoparticle. That's what we give the name called STEP. So it's clear. So it's not nanoparticle. So after this talk. So clearly, it's very efficient, and also Xiaona Lu is the one did all the work I'm going to show. Should remind me, I misspoken on the Thursday. So the timing for this technology is, I'm going to check it here, not here. So basically, it's the neuron from, this is a human neuron from the AS patient cell line. We generated and put a GFP in the tag so you can easily see that color. So basically, we saw the step, and then you can see almost like no expression in the UB3A, and it's a neuron derived from the Angelman patient cell line fused with the GFP on the paternal chromosome, the UB3A. With incubation of a step IMP in the within 24 hours, you can see beautifully reactivation, almost 90% of the neuron reactivate. So that illustrates how efficient this technology is. Clearly, we also show very, very nicely in vivo in the animal model, mice model. As Allyson said, we don't like a mouse model sometimes, but we do need some mouse model sometimes for different experiment. So this one shows you, even the 90 days after day 1, day 2 intrathecal injection, the spinal tap equivalent in the human, which is inject 1 dose of the step IMP, you can see clearly this is without injection. Here's the 0, no detectable. With injection, it's roughly you have about 30% to 60% depend on different brain region, the reactivation. So that's a pretty efficient based on all we learned from the mosaic patient Allyson talked about yesterday. So they are good, and of course you want to see if this indeed, I just kind of zoom in a little bit more kind of high power view, you can see this efficiency. It's amazingly efficient. Whenever we show this slide to our colleague, use this deliver technology, people are always amazed how efficient this single dose of injection. You get about 75% of neuron was edited by CRISPR, and then suppose these 76% of neuron, we have a reactivation of the UB3A gene. We are very, very excited for this technique for sure. And then, of course, you want to see whether this reactivation, like many other ASO and other approach, can rescue or correct the behavior phenotype, which is primary concern for many Angelman patients. So this is definitely a complicated slide and prepared by Xiaona Lu, the research associate. She's here. She gave a talk yesterday from the large delusion mouse model. And it's accumulated, this is one single slide. It's since last November or last December we presented. It's 1 year of work, a lot of work summarized this slide. So I won't be able to go detail for each experiment or result she has done, just show a few examples. Which is the first one, which you can see we call the open field. We basically put the mice in the kind of open field and look at their locomotion activity. So here's the wild type normal mice. Here's the mice with the Angelman syndrome. You can see the reduced of locomotion. Then after correction, day 1 of the intrathecal injection, you can see that pattern. It's very visible, very easy to tell. The kind of correction, almost close to the computer rescue for this particular behavior. Okay, that's the video, and I think I'm going to skip this one quickly. So this is second one, is the lower one. I think I'm going to just show you video. Allyson showed you a video yesterday, and this is a day 1, day 2 injection. Then I think it would be better for me to show you, okay, I cannot go, just skip this one because you saw this one yesterday. All right, so this is the one I want to show. Oh, sorry, can we play this one? Can we play this video? I cannot do here. So basically, this is the day 42 treatment. So shows you after, like day 42 treatment, still correct the lower function behavior as efficient as day 1 for this particular behavior. So that's a kind of important, but of course, this is the mice. A mice is a mice, and the human mice is not human. So that's make sure that's interpret data a correct way. But at least it shows the mice you can indeed later administration treatment can correct behavior phenotype, which is also true as a researcher community. We correct 100 different mouse models, but we also struggle in the human. So that's the difference there when we ever to. So we cannot play this one. Sounds like we cannot play this video. But then we also looking for the sort of intellectual ability of using multiple different testing. Again, this is a complicated slide with multiple replication. It's a beautiful replication. We have very high confidence this delivery working very efficiently, like a different time point, like day 1, day 21, day 42, and the different paradigm. This is a novel object recognition you heard yesterday. Basically, very quick it goes through, indeed, the correction of short-term memory versus long-term memory. And the short-term memory is very effectively corrected. Long-term memory, not too much. So that tells you that correction somewhere is a behavior domain specific, which is not a surprise. The last one I'm going to show is the seizure. Of course, this is one of the important features, or also oftentimes challenging feature for many of the family here. Your treatment for other medication may not be effective. So indeed, this treatment for step gene correction can suppress or correct the seizure behavior, seizure phenotype, pretty remarkably efficient based on the different treatment at different dose and different timing. I think I'm going to stop. Yeah, I'm going to pass to.
Okay, so this is really just meant to show the next step, which is how do we get from all that incredible work that showed proof of concept that in both a newborn, a young, and an adult mouse model, we can rescue almost all of the phenotypes of the behavior of the model, which gives a lot of proof of concept that we can actually take this and bring this to patients once we have a human candidate, so the human sequence in which we can employ this type of therapy. So we're at that point now, and what the next steps are is how do we get that from the mouse to the human? What are those steps? And those steps is what we term translational research. And so ultimately, I just want to give a quick description of that and really have you understand why it's so complicated and also why it's so expensive. So these steps are really what it takes to get an investigational human drug or biologic, so this candidate, that is declared from work in the animal model to humans through the regulatory authorities. Experiments that are meant to evaluate for critical information related to toxicity, target engagement, off-target risks, and overall safety for the human application. And these studies are referred to as IND-enabling studies, and we talk about that a lot, but let me just share with you what that entails. And ultimately, it involves many activities that are incredibly rigorous in order to get to that point, and we do them often in parallel, but by doing them in parallel in order to improve your timelines, it winds up costing a lot of money. So it is one of the most expensive parts of the drug development program prior to the clinical trial. So there's different pieces to this. One is called pharmacodynamics, and what that means is the drug working as you anticipated it would? Is it knocking down the paternal antisense transcript to upregulate the paternal UBE3A gene? The next is biodistribution or pharmacokinetics. Where does the drug go where is this candidate in the brain? Is it in the structures to the degree that we need it in order to be able to theoretically provide the benefit that you're hoping for? Can it turn on the paternal allele in all of the vital brain structures? And then toxicology, which is the dose levels you're testing safe? And that will drive the dose levels you can translate to in the clinic to ensure you have a nice margin of safety to say you're not going to go into the clinic with something that could have incredible risk. So what are all of the things that are needed to prove that it's safe? And then finally, are there toxicities called genotoxicities to the genome, to the chromosomes, to other genes around it? Is there an off-target problem? Will it change the genome in a way that's irreversible that we have to be concerned about? And these are all of the studies that we have to do in order to get to a human trial. So I just want you to see how complicated this is. And this is why things take a long time, even though we're all equally impatient. And so this is just an example of a drug development plan and all of the work that needs to be done to get there. And this is just to show you these arrows. One thing relates to another, relates to another. We have to do that. Before we do that, we have to do this in order to do this. And the only way we get to first in human is to be able to have all of these parallel pathways meet. And each one of these pathways has to have a go decision. So there's a chance at each one of these steps that there's going to be something that we need to pivot from. That doesn't make sense, that is not going to allow us to take this path forward. So this next huge step is really vital to understand if we have a path forward to humans. And so overall, studies that need to bring our human candidate to human application takes a lot of coordination, a lot of time, and a lot of money. We're obviously very careful as we make these coordinate these different types of studies, because they're critical, but we want to do them in parallel in the right way. And that includes the toxicology as well as the manufacturing, the regulatory component of engaging the regulators, and then the clinical trial pieces that we ensure we're running the right type of trial. And during every step, there are going to be unexpected findings. And that's why it's important to have a very nimble team that can work through those findings and ensure that we understand how to navigate them. And so we're really excited because we have a grant that's allowing us to take this work and leap forward from mouse to human. And the NIH is providing a large portion of the cost in order for us to be able to do this. Though there's going to be a lot more that needs to be done in this drug development process that they're not going to pay for, that we're going to have to really support if the decision is made to go forward. And so next, let's have Liz talk to us about how we're going to bring that information to a clinical trial.
Okay, so if these other parts are the Uber, I'm the destination. So what we're going to do, this grant is set up so that you get all of these other preclinical pieces done, and then we run a first-in-man clinical trial, which is going to be a pretty intensive kind of thing. And so I'm going to talk to you about what that might look like. The grant required that we study 2 diseases, or we couldn't get funded. So we are studying 2 diseases, and then we're going to kind of take them in parallel through this process. So we don't actually start until about year 3 of the grant when we start planning, and assuming that all of those other pieces go okay, then we can bring this to people. So we'll need to have an IND approval for the FDA, we'll need to have the protocol approved, we'll need to have our IRB approval, and then we'll no doubt, because this is a new kind of compound, we'll be enrolling adults first for safety reasons. We're going to try to run the trial at Rush and Yale, and we can train some of the Yale people in anything needed to do this trial in Angelman syndrome. We know this is going to be intrathecal delivery. We believe it's going to be intrathecal delivery. The location is probably based on preclinical data, and we could do this by LP infusion, but it could also be intracerebroventricular or ICM, as you heard about in the back of the neck. Whichever suggests the better delivery. Because again, this is one and done, and so you want to make sure you have your best delivery system for that at that time. And we expect a long-term correction. So this is what the trial design will kind of look like, and this is a sort of a standard design for things that are very new and in phase 1. We'll be dosing 1 patient at the beginning. We'll be running these 2 identical trials in Angelman syndrome and H1 syndrome, and then we'll have one patient, and then there'll be a safety period, and if that patient is safe, we can enroll 3 patients, and this will start with low dose, and then if these patients are all safe, we can go to the middle dose, and then the high dose, and then if a patient has a dose-limiting toxicity or kind of a bad side effect, we can expand and do more patients at the lower dose, and if 2 patients, say at this dose, had a dose-limiting toxicity, then we might have to go back a dose, or we might have to stay at that dose. So we find out what dose might cause problems. And then after we get adults done, we can move to children and do the same process with the children, where we enroll 1 patient and then we enroll more patients in a systematic way to make sure this is safe. And then, you know, it was fairly easy to write a design for what we want to do in this trial, because we have all these other trials going on in Angelman syndrome. And so we're going to largely use the same measures that we're using in the ASO trials, and we're going to have a lot of safety and tolerability kind of tests that we're going to have to monitor, and we can look at pharmacokinetics, much the same as all the ASO trials are doing. And I think it's important to point out that many of these measures were developed from ABOM data and natural history data, and there's the fast disease concept. So this work is all done ahead of time for us, which made it easier for us to get this grant. And so we have to study H1-4 disease, but fortunately that disease has a level of cognitive function pretty similar to Fragile X, which some people know that I've been working on for 20 years. And so we've already developed some nice, we've done some validation and adapted certain tests like the NIH Toolbox Cognitive Battery and the KITAP Executive Function Test and expressive language sampling for Fragile X. And we were able to make the case to kind of roll those over to use them for this other form of intellectual disability. And this kind of work where we adapt appropriate outcomes and trial designs and accelerate new therapies into patients by getting those early trials done for Angelman syndrome and neurodevelopmental disabilities is really the mission of our Rush Pediatric Neurosciences FAST Center for Translational Research. And this new grant capitalizes on the fact that we have the infrastructure to do this kind of thing. So it really highlights the point of the Center of Excellence. And then after that initial study, we can bring in more sites. If it looks like it's very successful, we can bring in more sites, train others, and hopefully there'll be a next phase of development of this kind of novel therapy. And that, so in conclude, this is really a perfect example of how supporting the infrastructure and collaboration allow us to make scientific progress. It brings teams together with various expertise to allow efficiencies that couldn't be achieved in any other way. And the work that we've done in the past to create what we needed to get this really allowed us to get this grant. And I think FAST really works hard to ensure that that we are the low-hanging fruit for these platforms to get funding from either industry or NIH to look at these kind of proof of concept early trials and have the clinical capability to move it forward. So thank you to everyone. NIH, FAST, Yale, this is going to be a really important collaboration. See if there are any more slides. Okay, and then the acknowledgements. We have all of the groups that are working together on this, but we've already talked about that.